Technical Veterinary Learning TVL channel, founded and produced by Professor Ahmed Mamdouh El-Sharif, Professor of Epidemiology and Infectious Diseases of Animals, the ex-head of Department of Veterinary Medicine, the ex-Vice Dean of Faculty of Veterinary Medicine, Bani Suef University. We will speak now about the viral diseases of camels, management of diseases outbreaks in livestock farms. Viral diseases of camels. Camel Rabies Disease. Rabies is a sporadic, acute, highly fatal viral infectious disease infecting all warm-blooded animals and zoonotic in nature. Rabies disease generally characterized by encephalomyelitis, abnormal behavior, progressive ascending paralysis, nervous manifestations differentiated according to species and usually ended by death. Rabies disease is an OIE-listed lethal zoonotic encephalomyelitis disease and a major challenge to public and animal health that causes thousands of human deaths yearly worldwide. Developing countries in Asia and Africa bear the heaviest weight and account for 95% of human rabies cases. This is reported by WHO 2020. Rabies is known by a highly variable incubation period and a high case fatality rate, as it almost fatal upon the appearance of clinical symptoms in both animals and humans, when there is no proper post-exposure prophylaxis. Like all warm-blooded animals, camels are susceptible to rabies disease. Rabies has been accounted for in dromedary camels in all camel-raising countries. Growing camel farming, stretching into the desert or bushes increases the chance of frequent attacks by wild carnivores. Rabies is believed to be present in every country in which camel husbandry is practiced. Pathogenic risk factor. Rabies is a viral disease caused by rabies virus, which is negative sense single-stranded RNA virus of the genus Lysovirus in the family Rabidoviridis and order Mononegavirales. This figure shows the shape of rabies virus Lysovirus family Rabidoviridis 75 multiplied by 180 nanometer in diameter, like pullet. The rabies virus represents taxonomic prototype species. Rabies Lysovirus is enveloped and has a cylindrical morphology, pullet-shaped. Lysoviruses are normally limited to one major reservoir species in each geographic area, although spillover to other species is common. Rabies virus has been isolated from nearly all mammalians. Identification of different virus variants by recent laboratory technique such as monoclonal antibody analysis or genetic sequencing, they has greatly enhanced understanding of rabies epidemiology. Herbivorous including camels and omnivorous like man are the final hosts, while bats are considered the sources of the virus, either carnivorous and insectivorous bats. Rabies virus is sensitive to 1% formalin, 3% cresol, ultraviolet rays with 245 nanometer wavelength, boiling 70% ethanol soap solution. Rabies virus is sensitive to also chloroform solution, ether, quaternary ammonium, acetone, 1% sodium hypochlorite, 2% glutaraldehyde, iodine preparations. Geographical distribution. Reported cases of rabies in camels came from United Arab Emirates, Oman, Qatar, Saudi Arabia, Morocco, Niger, Mauritania, Nigeria, Sudan, India, Kazakhstan, Iran, and China, as in the following map. This figure showing countries report occurrence of rabies in camel, taking red color as we said before. All camels at different ages are susceptible to rabies disease. Source of infection to rabies disease including infected dogs and infected jackals. Mode of transmission to rabies disease. Rabies virus is usually transmitted to the next host by bites through the saliva of a rabid animal, but also via scratch wounds of contaminated mucus. In most cases, the disease is transmitted by bites from rabid wild dogs and less commonly from bites of rabid foxes and cats. However, other animals such as wolves, jackals, hyena, monkeys, or mongoose could be transmitted the disease easily when they are infected. Camels are bitten commonly on fore or hind limbs and in the head of females when they defend their neonates from predator attack. This figure showing 4 transmission cycles of rabies disease between livestock animals and carrier animals. The first one, rabies virus circulating within pets communities, where fox, dogs, skunk, and raccoons are the main vectors of rabies into livestock. This reported in the Northern America and Europe. Second cycle, when rabies virus maintenance within terrestrial countries, where dogs and jackals are the main vectors of rabies into livestock in Africa and Middle East. The third one, when pets, foxes, and dogs are the main vectors of rabies into livestock in Latin American and Caribbean countries. The fourth one, when rabies virus can infect all livestock animals including camels, where dogs, jackals, and raccoons are the main vectors of rabies into livestock in Asia and Africa. Pathogenesis of rabies disease. The incubation period in most animal species varies between 15 and 30 days. This diagram showing the pathogenesis of rabies disease in dogs. When biting by another carnivore, the virus will replicate at the nerves and muscles, then spreading through motor nerves to become replicating virus in the spinal cord, which reach to the brain and then replicate in the salivary gland, where saliva here become infectious. Pathogenesis of rabies virus in livestock animals. Rabies virus in saliva of infected dog, when it bitten the forearm or hind arm or the head of any domesticated animal. The rabies virus in the bite site enters the motor neurons in the plate, and then the rabies virus will replicate it in the muscles. So it will reach to the peripheral motor nerve. Then the rabies virus multiplied in the spinal cord. From the spinal cord, the rabies virus will be coming in the brain, and then will enter the salivary gland and replicate it, and the saliva become infected with the virus and as a source of infection. This figure showed how she camel bitten by an Arabian red fox. All camelids are theoretically susceptible to rabies virus, but most of the available publications described clinical rabies in dromedary camels, with few reports in bacterian camels, Camelus bacterianus. Reported that camels are one of the important sources as well as carriers of rabies infection for human, livestock, and wildlife and worldwide. Clinical picture of rabies in family Camelidae. Rabies disease in camels is mainly of the silent or dumb type. Various form has also been noticed, particularly in males, in which affected animals become excited, aggressive, attack and bite any nearby objects, as well as self-biting of forelimbs, and become self-destructive by pushing into and over obstacles. The early clinical signs of the disease include restlessness, ataxia, posterior paresis, salivation, and rotation of head and neck in all directions. These signs are soon followed by paralysis, recumbency, and death. Other signs that have been observed include bloat, pruritus, muscle tremors, facial paralysis, aimless running, sexual hyperactivity, spontaneous ejaculation, recumbency, convulsions, and coma. The clinical course can be divided into three general phases. Number 1, prodromal. Number 2, acute exudative. Number 3, paralytic state. During the prodromal period, which lasts 1 to 3 days, animals show only non-specific signs which intensify rapidly. The disease progresses rapidly post-onset of paralysis, and death is virtually certain a few days thereafter, and some animals may die rapidly without marked clinical signs. This figure showed a rabid dog with various form. This picture showed rabid dromedary camels with yawing. Yawing in rabid dromedary camel due to esophageal paralysis. Rabid dromedary camel showed jaw paralysis. A rabid dromedary camel showed various form. Salivation in a rabid bacterian camel due to esophageal paralysis. Salivation in a rabid bacterian camel due to esophageal paralysis. A dromedary camel affected with rabies showing lateral recumbency and excessive salivation. Zoonotic importance of rabies. Rabies is a lethal zoonotic disease. Laboratory diagnosis of rabies disease. Samples collection. Number 1, from live animals, saliva in closed package and sent directly to lab before dryness. Number 2, from dead animals, brain of dead camel, where according to OIE laboratory techniques are preferably undertaken on central nervous system tissue removed from the cranium, for example brainstem, ammonas, horns, thalamus, cerebral cortex, cerebellum, and medulla oblongata. From dead animals, a composite of central nervous system samples should be tested, and the brainstem is the most important component of the sample. Laboratories should follow appropriate biosafety and contaminant procedures as determined by biorisk analysis. Fresh brain of the camel should carefully be collected into ice and immediately transported to the laboratory. Number 1, histopathological examination is a safe and reliable diagnostic tool in rabid dromedary camels when only formalin-fixed and paraffin-embedded material is available, but negative results should be confirmed by direct fluorescent antibody technique or RT-PCR. This figure shown negri bodies in the brain of a rabid camel. Intracytoplasmic negri bodies in a brain of an infected animal. This figure showing intracytoplasmic over eosinophilic inclusion bodies as negri bodies as seen by arrows in a pyramidal neuron in the cerebral cortex of a rabid camel. Scale bar 1/4 20 micron. Number 2. Currently, diagnostic techniques have been standardized internationally that includes the following according to OIE 2019. Number 1, direct fluorescent antibody technique test. Number 2, direct rapid immunohistochemical. Number 3, pan-lysavirus polymerase chain reaction PCR assays. Direct fluorescent antibody test is the most widely used reference method for rabies diagnosis. However, results must be read by an experienced kill analyst already vaccinated against rabies. This figure showing direct fluorescent antibody test showing rabies antigen as small green particles in the brain of rabid camels, and this directed by the yellow arrows in the figure. This figure showing the direct fluorescent antibody technique test detect rabies antigen as small green particles in the brain of rabid camel. We magnify the picture here. In cases of inconclusive results from these diagnostic tests, further confirmatory tests by RT-PCR, cell culture, or mouse inoculation tests on the same sample or repeat primary diagnostic test on other samples are recommended. Wherever possible, virus isolation in cell culture should replace mouse inoculation tests. This figure of agarose gel for diagnosis and the detection of rabies virus genome by RT-PCR amplification noted at 586 base pair at the lanes 2, 3, 4, 5, and 7. This is a phylogenetic tree of N-gene nucleotide sequences of 2 rabies virus by Nepal joining method. Differential diagnosis. Rabies can easily be confused with other diseases or with normal aggressive behavior of animals as following. There are 7 diseases with nervous behavior have to be differentiated from rabies disease in camels. Number 1, meningeal worm and abscess. Number 2, listeriosis. Number 3, tick paralysis. Number 4, copper deficiency. Number 5, camel prion disease. Number 6, normal male sexual behavior, rutting during sexual season. Number 7, cerebral synuresis. Treatment of rabies disease. As there is no effective treatment for rabies, any confirmed rabies case must be killed immediately and its carcass incinerated or burned. In the field, a rabid camel showing clinical signs is usually segregated from the herd and left to die in the desert or destroyed when aggressive. Post-exposure prophylaxis protocol by rabies vaccination should be implemented as soon as possible in a timely manner after exposure. However, there is no reports about the use of vaccination in camels. Control of rabies disease. Once rabies is diagnosed in a farm, the veterinary authorities thought to be promptly implement the appropriate control measures that include: number 1, incineration or burial of all infected animal carcasses, emergency vaccination of dogs in the area. Number 2, raising of public awareness. The elimination of stray dogs and other animals by shooting and poisoning is still implemented in certain countries. However, this minimally affects rabies transmission. Certain countries of the Middle East region are converting expanding problem because of wildlife rabies. The red fox and golden jackals are examples. Prevention of rabies disease. Measures to protect camels from predators like stray dogs, foxes, and hyenas are fundamental for prevention. The World Organization for Animal Health (OIE) has recommended rabies prophylactic vaccination for companion animals and livestock using inactivated virus. However, rabies vaccination in camels has barely been reported. In recent studies, a single injection of double-dose canine inactivated rabies vaccine was found effective and conferred protective immunity in camels for at least 1 year, with booster dose every 6 to 8 months to have acceptable level of protection. In any case, successful control and eradication of rabies are accomplished through effective vaccination program for pet animals and wildlife, which is generally difficult to achieve in most developing countries. This is reported by OIE 2019. Notification. Rabies in all animals is untifiable disease. According to OIE, camelpox is untifiable disease. We keep on the track. Did you? Please subscribe the channel to reach to the most newest knowledge about the different infectious diseases of livestock of animals. We keeping you on the right track. So please subscribe the channel to reach to the most newest knowledge about the epidemiology and different infectious diseases of different species of animals. See you later.